will talk about yet another area of his research, is to apply machine learning for protein RNA complexes. Hopefully we'll hear about the lessons learned after the last round of CASP, and I would love to ask him for his comments on how to improve deep learning methods for more accurate RNA predictions. And with that, the screen is yours, Frank.
All right. All right. Yeah. So thank you again for the invitation. And I mentioned this. I want this to be interactive. So if you have questions, please ask them throughout the talk. Just maybe raise your hand in Zoom, and I can call you out. I've left lots of time. I've only probably about 30 minutes of talk prepared. So yeah, I'm Frank DiMaio from the Institute for Protein Design, and I'm going to be talking about the work that my group has done, basically trying to use, to add to RoseTTAFold, to add to this machine learning guided structure prediction, the ability to predict nucleic acid structures alongside of protein structures. So of course, this is a machine learning guided method. I'm going to give a little bit of an introduction on machine learning guided methods, kind of, and the developments leading up to AlphaFold. This is probably review for most people here, but I'll go through this relatively quickly. So, so I think the idea of machine learning guided methods is kind of built on this idea of sequence coevolution. And so the idea here is if you have some protein sequence that you're trying to predict, and you don't know anything about the three-dimensional structure of that protein, but you have the sequence of many, many homologous proteins, you can identify, let me just get a pointer here, just so, yeah, you can identify positions that essentially mutate together, these so-called co-evolving sequences. And people a while ago, more than 10 years ago, recognized that if you have enough sequences, again, then you can use this to predict. Essentially, on the right here, I'm showing a three-dimensional contact map of the protein. These are residues of a protein that are in contact with each other, that you can predict these pretty accurately. In fact, again, given enough sequences, you can predict this accurately enough to determine the three-dimensional structure of a protein overall. And so, this was really powerful, but of course, you need many, many sequences. A lot of proteins that are interesting didn't have enough sequences. And so people, I guess around 2017, started to realize that you could approach this as a problem of computer vision, of like image denoising. You could think of these contact maps as two-dimensional images. When you don't have many sequences, these are really noisy images, and you can use ideas from computer vision. And specifically, so a number of groups did this. I think Jimbo Zhu's group was the first that approached this, but a number of other groups, including the original AlphaFold group, basically took this coevolution signal as input information into these kind of deep convolutional neural networks. We're able to predict contact maps, or able to predict distance distributions between residues, fold subject to that. And this dramatically, increased the accuracy. And then, of course, as people are well aware, in 2020, AlphaFold 2 came along and really made kind of this huge leap forward in terms of performance. And to just show kind of the differences in what AlphaFold and what the DeepMind team really introduced, I'm showing a comparison here between, this is TR-Rosetta, which is just one of the standard convolutional neural network based structure prediction methods, again, similar to the original AlphaFold, or similar to the code from other groups as well. And what I'm showing is just sort of a cutout from DeepMind's talk from the CASP 2020 meeting. And I think there are a couple of key differences here that really kind of led to this performance improvement. The first was sort of the machine learning architecture that they used. TR-Rosetta and previous methods had used these convolutional neural networks, which are very popular in computer vision, where DeepMind made use of these attention-based mechanisms, which are more popular in language. And kind of the difference between these two has to do with how information flows. In convolutional neural networks, information in an image is going to flow at each level to nearby pixels. It makes sense for images, but for proteins, where your pixels are residues in the protein, this doesn't really make a ton of sense. So if the N and C termini are near each other, you don't want to have that information take forever to get from the N to the C terminus. And so I think that was another piece. And then the second piece is that previously groups had really focused on kind of separating the problem of contact map prediction and structure prediction. And so the idea is that you can predict the contacts of a protein, and then you can use kind of traditional physics-based refinement to then build a three-dimensional structure that's consistent with this predicted contact map or this predicted distance distribution. And what DeepMind realized is that you can actually have the neural network itself reason on the three-dimensional structure of the protein. And this offers, I think, a couple of distinct advantages. I think the first of these is that it lets the network learn that this kind of these 2D features, this contact map, that its prediction actually has to correspond to a realizable three-dimensional structure. And again, having that information flow back from the structure module to kind of this 2D information, this 2D predicted contact map is really powerful. And then secondly, I think a real powerful thing is that it allows sort of uncertainty information to flow all the way through the network as well. So what we did basically, so following this 2020 CASP performance, so Mink-Young Baek in David Baker's group really spearheaded this, but basically from these principles, tried to incorporate these principles into a kind of a novel machine learning architecture. This was before AlphaFold, the details had been released, and this was released in 2021 as RoseTTAFold. And so what Mink-Young had come up with was basically the model architecture that's shown here. So AlphaFold, as I mentioned, has sort of two different tracks. So there's a track where they're reasoning on sequence information, and then a track where they're reasoning on contact information, and then that feeds into a model that uses that information to predict 3D structure. Mink-Young found that by also including a track that reasons on three-dimensional information, that basically required fewer parameters for the model, and it seemed to perform better. Additionally, so basically this was in 2021. I don't want to go too much into the details, but subsequent to this, Mink-Young and I have been continuing. So Mink-Young has her own group now, and her and I have been continuing to develop RoseTTAFold. And specifically, we've been trying to incorporate different features into RoseTTAFold 2, trying to push this architecture to kind of the best performance possible. So compared to AlphaFold, there are a couple of key differences that I wanted to point out. So first of all, again, obviously the presence of this three-dimensional track. So throughout the different layers of the network, we're modeling, we always have an idea of what the three-dimensional structure is at a given point, and that three-dimensional structure is used to update the 1D and 2D tracks. In terms of the kind of the machine learning architecture, there are a couple of key differences. So instead of the sort of invariant point attention that AlphaFold is using, we're instead using this SE3 transformer. And again, we've continued to use that. And then secondly, we again, AlphaFold made use of this this novel kind of machinery, this triangle attention that seems to work very well along the 2D track. And so what we decided to use instead is that we found, so using basically this biaxial attention, but because we have the structure track, we can use the three-dimensional structure to actually bias the attention in the model. So basically teaching the model that it should be passing information, that attention should be highlighted on residues that are near to each other. You can use the structure of the triangle attention as trying to capture that, but also, again, the 3D track itself, information from the structure can feed into that. And this is really nice because this is a slow order L-cubed operation in AlphaFold. Instead, this is just an L squared. You're just doing attention across the rows and then attention across the columns. Those are the major differences. There's a lot of like small details. And anyway, so we've been working on this, subsequent to the publication of the original RoseTTAFold model, and now the performance of this seems to be in line with AlphaFold. So I'm showing, so this is just on a set of very recently solved structures. I think this is around 400 structures that were solved in late 2022 and early 2023. And so what we're comparing here is both on monomer structure prediction and on complex structure prediction. We're comparing the GDTTS of the original RoseTTAFold, AlphaFold 2, and AlphaFold Multimer. And you can see RoseTTAFold 2's performance on monomer structure prediction is in line with that of AlphaFold. Rosetta's performance, the same exact model on complex prediction, is in line with AlphaFold Multimer. And like I said, because we're not using this triangle attention, instead we're using, Instead, we're using still this just biased biaxial attention. We see basically L squared instead of L cubed scaling in terms of protein length. And I'll talk more about this in a second, but basically when proteins get large, particularly 1,000 residues or larger, it is significantly faster to predict the structure. And then finally, I guess the last thing that I wanted to show is on the right, kind of showing the development over the past year of this model and the different features that we added. And a lot of this was just really clever engineering that the AlphaFold team had done. They did some really clever things, and incorporating those in the context of the original RoseTTAFold model helps a lot. So for example, fate loss that they used, they had this sort of frame aligned where you basically, rather than trying to do whatever singular value decomposition to get your optimal alignment, it just kind of considers each residue pair, each residue's frame, and computes the coordinate loss. This worked great, and that alone, just changing our model to have that fate loss, again, led to a huge performance improvement. The idea of recycling, where your network outputs feedback into the inputs and using them in training was also, again, another valuable thing. And then other than that, a lot of small changes, increased network depth, the idea that they're using very wide MSAs for the first couple layers of the network. And most of these ideas, I think, do come from AlphaFold and really show kind of really the great engineering that was part of that. But we also show here that, that some of these architectural features like IPA or like triangle attention aren't really necessary to get the high performance of AlphaFold. And so that's really great. Of course, this is something, so AlphaFold did this 2 years ago. What's so special? So I think what's neat is we understand, at least to some extent, how this works now, and we can start doing new things with this network. And so just briefly, in protein space, I'll talk about this, and then I'll get to the kind of nucleic acid work that we've been doing that I think this group is probably more interested in. So this network, it's a lot more efficient, and we can predict really large proteins now and really large complexes. This just, for example, is a 4,200 residue protein with 1,024 sequences, pretty big MSA. It runs no problem, about 15, 20 minutes of runtime. And there's more, we can run up to, on sort of these very high memory GPUs, we can run close to 5,000 residues. Times of inference, which is really nice and relatively quickly. The other thing that we've been really interested in is prediction of structures of complexes with known symmetry. A lot of times, the symmetry of a complex is known, and you want to predict it, and particularly if it's high order symmetry, predicting that accurately can be challenged. And so basically what we've done is take advantage of the fact that, so the slow step in both of these networks is going to be updating sort of this 2D track, so the residue pair features, making those consistent. And you can draw off the fact that there's a lot of redundancy if you have known symmetry. There's a lot of redundancy in this contact map. And specifically, I'm showing for a C5 symmetric complex here, I'm showing the contact map. And you can see that to build this entire contact map, you assume that this has C5 symmetry. All you need is this sort of patch. So basically this patch that represents one subunit interacting with all other subunits, right? And so by internally storing that and then further reducing things by only considering the subset that are interacting with your main subunit and updating that throughout the course of the network, you can really model symmetric complexes very, very efficiently. And so this is still sort of work in progress, but we're able to build, in this case, we're able to predict large symmetric structures, showing a variety of different symmetries and sizes, and even sort of large icosahedral capsids, we're able to predict, with a reasonable degree of accuracy in some cases. So further testing on this is going on, but this is really promising. And again, these cases are just too big. These 15,000 residues are just too large to predict asymmetrically. So, but the other thing, and so the main point of the talk today has been our work predicting adding nucleic acids to this network. And so we had this network kind of trained, and the architecture was pretty well worked out. And so the question was, could we actually then use this to predict nucleic acid structures? And so could we use this to predict both RNA structures, but also protein nucleic acid complexes? And so in terms of the engineering of this was relatively straightforward. There were a couple of new things that needed to get added to the network. They're highlighted here with these red arrows. Maybe I'll just jump to the next slide. But so basically implementing this, we needed to add just new tokens to the network. So the network, the alphabet that the network understood is the 20 amino acids, an unknown token, and a mask token. And we added to this tokens for the 4 DNA bases, the 4 RNA bases, and then unknown DNA, unknown RNA. It was relatively straightforward. The model, of course, is reasoning on structure, right? And so we expanded this structure model to deal with nucleic acid bases as well. And so specifically the way that RoseTTAFold's structure prediction works the same exact way as AlphaFold, that you predict a frame for each residue, you predict a coordinate frame, and then you predict a set of torsions that allows you to basically build, so they correspond to the rotatable torsions of the amino acid that allow you to build an all-atom representation of the amino acid. DNA and RNA, we can do the same thing. We chose as the coordinate frame the phosphate group. And so then basically there are 10 torsions that describe how to build all of the atoms of the nucleic acid. And those are the sort of the 6 backbone torsions, the 1 nucleoside torsion, and then 3 additional torsions that describe the conformation of the ribose ring. And we found that. Yeah.
Maybe I would ask a question, and maybe this will also encourage.
Yeah.
So have you considered using other frames? What was the?
We have not. So one of the things is it's an arbitrary decision. It's not totally clear how important that frame is. I know other work has used sort of the connection between the backbone and the side chain as the frame. I don't know how much of a difference that it would make, and it's challenging. And you'll see in a second. Training this network from scratch takes a very long time, and so it's not straightforward to run that experiment, I guess, is what I'm saying. I really don't know how much of a difference that the frame makes. And the frame is, I mean, in some sense, the frame is, the frame, in some sense, it's really important because it's the internal representation. On the other hand, like the loss is really ultimately driven by like the all-atom, by the all-atom representation of the structure. And so the choice of frame in computing your final loss isn't so important. Where it is important is like, again, the internal representation of the structure. I am not sure how important that frame choice is. It's definitely something to explore. But yeah, it's a great question.
Okay. Yeah. There is one from Hani. I don't know if Hani wants to say it. I can also read it. Is it possible to replace the contact map generated using coevolution with one generated using experimental chemical cross-linking?
So that's a great question. So that's something we've been interested in. I wasn't going to talk about this today, but one of the things that we're really interested in is being able to feed the network information, prior experimental information, for I think 2 different reasons. I think one of the reasons is just to aid in prediction, especially as you'll see here, these networks aren't as accurate as the protein-only networks. And then secondly, ultimately for understanding alternate conformations. If you could steer the network, if you know from experiment that 2 pieces are coming in close contact to one another, then you can steer the network into that state. That's something I think that is doable, but that's still very, very in the very early stages, I think, at this point. But that's definitely something that we're looking at. And so the idea would be to essentially when you train the network, to with that information sometimes available. So yeah, the other thing that we did that was new is we knew this is like limited data, and so the bottom talks about that, but we have less than 1/10 of the data. So compared, we went to the PDB, our training set itself consists of 17,000 protein nucleic acid complexes, but if you cluster these, a lot of redundancy, it's only 1,200 unique clusters, 6,500 RNA structures. If you cluster these, it's only 1,400 unique clusters. And so this is really not a lot of data. Compare this to, again, the almost 20 times as many protein-only clusters. There's, again, 300,000 chains in 21,000 clusters. And so one of the things that we tried in this was to actually include some limited physical terms in the loss function. So when fine-tuning the network, AlphaFold made use of just a clash loss. Instead, we actually put a proper, I shouldn't say a proper, but we put a 12-6 potential in for the Leonard-Jones loss. And then we also add Rosetta's hydrogen bonding term as an additional loss term to kind of favor, again, structures that make good hydrogen bonds. These weights are still really low relative to overall structure loss, but again, in the fine-tuning network, it seems to help. Yes, Rachel.
Okay, I have a question, and then Riju after me, you should unmute.
Yeah.
Say your question. So I'm asking about the clusters. Is this sequence-based clustering or
Yes, this is all sequence-based clustering, so it's not structure-based. So in terms of structural diversity, there's certainly even less than this. This is all sequence-based clustering.
And then Riju,
From Riju.
do you
Oh
want
yeah, I
to
wanted to
unmute
see.
and
Hey,
your
Rick.
question?
Hi.
I wanted to see. Hi, I wanted to see if you that you showed all these tricks for improving RoseTTAFold 2 on the previous slide. Are they also things like using the FAPE loss? Are those also being used for the nucleic acid?
Yeah. So basically, I think everything here is being used. All of these improvements are being used now in RoseTTAFold 2. Sort of as these were developed in parallel, so it kind of plugged along. But the current available version of RoseTTAFold nucleic acid does have all of these features in it.
Okay, then I think the follow-up question is that are you using the phosphate frames for the FAPE loss, or is there something?
Yeah. So, but again, the FAPE loss. So for the sort of, there's two different losses. There's two different ways the FAPE loss is used, right? So in basically the 40 structure layers itself, the FAPE loss is being used as just with the backbone frame, which would just be over the phosphate group. But then the sort of most of the weight, right? So essentially, each of the individual layers have like something like 1/40th of the weight. So you basically take the average of the FAPE over all the layers and then take the average of that with the final FAPE. And that final FAPE is over all rotatable, you basically build a frame on every single rotatable bond. So it's like an all atom FAPE at that point. And that's really driving. So that's why I don't think the loss is really driven by the but by the choice of frame very, very much. I think it is the internal representation that matters, because I do think the backbone, again, the weights are so light on the backbone only FAPE part of this. And that's something that we could potentially change very easily in test if that's making too much of a difference. Cool. Yeah, what would you recommend for the frame, I guess? Like what's
I think the problem is that with experimental structures from crystallography, and also, increasingly from cryo-EM, the phosphates are the backbone is really poorly determined. Yeah. And so I don't know, like 20 or 30% of, let's say 10 to 30% of the nucleotides in a given RNA structure will have like mistakes in where the atoms were positioned in the history.
Okay.
It's gotten a little bit better over time, but like it used to be really bad. So I think like base frames are more robustly determined.
Okay. So yeah, that's
like
something.
It's an easy update. Yeah.
Yeah, that's something in terms of loss. That's something that's very, very easy to put in terms of like the actual generation of structures. That's where it's challenging. And that's why ultimately like the 2D, right, the 2D representation completely changes in that context. And so, but I do think that's something that we could definitely play around with.
Got it. Thanks.
Yeah. And then, so then what are we doing? So with training, again, we're taking basically proper RoseTTAFold training. And then we add to it two different kind of sub-training tasks. So protein plus nucleic acid, and that includes both RNA and DNA structures, and then also RNA prediction. And so the RNA, we use MSA information, and we used, again, just some sort of prior, based off some prior publications, but essentially, again, just searching NT and then searching this RNA Central database, again, to build MSAs for RNA. And then for protein RNA complexes, we use we use a trick where basically, you compute the MSA for both chains individually, and then you kind of combine them together using taxonomy IDs. And this was the same trick that's used in predicting protein complexes, basically. And so, yeah, so this was sort of our first. And when we first started doing this, I had no idea like what would work, what wouldn't work. We actually tried. We weren't sure that the nucleic acid would work at all. And so we had originally tried to with training this with templated nucleic acid structures. So I think originally 25% of the time, the perfect nucleic acid structure was given to the network, and it was just trying to predict how a protein folds on that for complexes itself. But it turns out the network learns this, I would say, reasonably well. So I'm showing the early training curve of RoseTTAFold 2. This is the line in blue shows protein training. I'm just showing all atom LDDT, accuracy measure, higher is better. RoseTTAFold 2, the data that I just showed, it eventually hits 0.82 is the value we get to. This is still early training, and so this continues to improve. But you can see relative, and the main reason I'm showing this is you can see relative to this, the performance of RNA-only structure prediction, and then protein nucleic acid. And from scratch here means we're building everything from scratch. So in this case, we don't give it any nucleic acid information. We don't give it any protein information. It has to build the complex from scratch. So I'm going to talk now about some of the results. And I wanted to start with the RNA results. And then I'll move on to the protein nucleic acid complex results. And that's where I think unfortunately, most of the development has been on the complexes, but I would love to get some feedback in terms of the RNA. So the results are shown here. So what we did to evaluate this network is that we basically stopped training the network. Sorry, our training data was limited to, I think, mid to late 2020, data from mid to late 2020. And so what we did when validating the network is we take all of the data, all of the RNA structures that have been published newer than this, and also all of the RNA structures that were in the validation set for training, all right? And so then what we did is to compare how well this prediction is, we compared essentially whether the sequence similarity was close to anything that the training set had seen. And that's what we're showing on this plot. So the x-axis, basically using BLAST-N, the E-value of the closest structure in our training set. So all of these structures on the right have basically, in terms of sequence identity, nothing that is identifiable. The structures on the left, again, everything kind of to the left of this line, there's basically some sequence similarity to something in our training set. And what you're seeing, again, LDDT here. So what you're seeing is the network. So on one hand, the network isn't super accurate. We get a success rate, again, depending on what you're considering a success. You consider, say, maybe 0.7 LDDT a success. We're seeing success rates of around maybe 40%, so 40% of structures. But one of the, again, powerful things about this is the network predicts its own accuracy. And that's what the colors are representing in this plot. And so what you can see is that the network, if you focus only on high confidence prediction, then the network is very, very accurate. So if you focus on predictions where, say, the predicted accuracy is higher than 90%, so that you basically can only accurately predict, 30% of your structures, but of those 30%, they're all essentially very, very accurate structures. So that's pretty powerful. And then on the right, I'm showing some examples of this, and these are examples that have no sequence homology. So all of these examples, unfortunately, do have structural homology to something that's already been solved. And with RNA, I think there's only so much diversity that's really apparent. But all of these are novel, at least in terms of like BLAST cannot detect any sequence similarity to anything that it's seen. And you can see, again, 4 different examples of it. And again, you can see sort of some of these cases, again, some of the base flips. So I'm showing the native structure, in this case on the left or above, and then I'm showing the predicted structure on the right, and then the gray, the kind of shaded outline here is showing like the superposition of the two. So we also, again, this is obviously the CASP group, we also entered this in CASP, and I think as Riju pointed out in his recent preprint, these machine learning guided methods actually didn't work very well, and our method just generally didn't seem to work particularly well. I'm showing some of the CASP 15 targets here that we predicted. And so basically what I'm showing on the top is the best server method and the best overall method compared to RoseTTAFold. This is just showing LDDTs. And again, you can see we're really falling behind, particularly the best human group. And then I'm comparing to 2 different machine learning methods. And so again, the results are sort of more in line with this, but you can see that we're still doing a little bit worse. I think the reasons for this, I think there's 2 features that we're missing. I think these other machine learning methods make use of this idea of like a distillation set that I'll get to in a little bit. But basically taking network outputs and using that to kind of beef up your training data set. We haven't had the chance to do that yet. That's something that's definitely on our to-do list. And then also kind of tailoring both your loss and also giving the network input features that maybe provide some additional kind of prior knowledge on structure. Like, so for example, giving secondary structure predictions as an input, for example, might help the ability of the network to predict these structures. So that was basically RNA. And so then we were like, yeah, go ahead.
Sorry. But yeah, we have a few questions and comments. So my question would be, so what kind of alignments you used for CASP? They were the same alignments that you would obtain now getting the RoseTTAFold from GitHub?
Correct. Exactly.
Mm-hmm.
So it searches, like I said, it searches different databases basically, but the big ones are the sort of NT database, and then it searches RNA Central database and builds the alignment. And yeah, it uses, I think it kind of follows what we're doing. Oh, it kind of follows what we're doing for proteins, right? Where we do kind of more and more permissive searches until we get an effective number of sequences, basically, yeah.
And the network itself was the same model that it's now on GitHub, or like in terms of parameters and the details?
Yes. You mean, so in terms of this data, so the data we actually submitted as part of CASP was an older version of the network. This is a sort of rerun of all of that data. Yes. This is a rerun with what currently is available.
Okay, and there is one comment that it seems that the tool will not work only for RNA if you don't download lots of data for protein sequences, but I'm not sure.
Oh, that shouldn't be the case, but I can look at that. If you can make a comment, if people are curious about using this, if you can make a comment on the GitHub repo, I can definitely address those things. That shouldn't be necessary. The protein, so for RNA only, it shouldn't be necessary to have the protein databases downloaded.
And there is,
so that can definitely be fixed.
Okay, from Summit, the installed model.
Oh, so Riju, oh, sorry, go ahead.
Okay, I just want to go from the top, but yeah, we'll go to Riju. The installed model provides slightly different results in each run for protein structure prediction. Is this deterministic?
So no, the model is not deterministic, and the reason for the is that basically we initialize structures into a random cloud of structure, a random cloud of residues, and that gives to some amount of stochasticity to each run, and it really does depend upon like the certainty of the prediction. In cases where the predicted LDDTs are low, where the network is unsure, it will often give you very, very different predictions each time you run it. In cases where the predicted LDDT is high, generally it will give you almost exactly the same prediction each time.
And now I see that Sergey actually commented.
Yeah, Sergey answered that as well. Yeah. And also, yeah, so that it's the random cloud. There's also, if you have big MSAs, at least there's MSA subsampling also that goes on. If your MSA is bigger than what the network can handle, it also subsamples at each iteration different subsets of the MSA, and that also leads to different things. Yes. And then the PLDDT. So Riju asked about the PLDDT. In these cases, the PLDDTs I think were uniform. I don't have the exact numbers, but the PLDDTs were fairly low. They were in line with the predictions themselves. Like we see pretty good correlation between PLDDT and LDDT. So we kind of knew that these predictions, the network didn't know them very well. These were also, I think many of these were, I guess Riju probably could comment more than I can, but these are a lot of these I think were designed structures, and they had all very shallow MSAs, at least when we ran them. And so that again generally leads to, unless the RNAs are very, very small or very, very clear, that often leads to poor predictions. And so the network knew that these predictions weren't very good, basically.
And I think there is one comment in terms of the data set. I think Summit asked if it would be possible to upload data sets, at least a list of containing training and test. So I believe the list of
yes, yeah, we can do that. Yes, and so like putting up the training scripts has been like on the to-do list. It's a little just complicated in terms of getting the code in a state. It's right now tied together with a bunch of scripts. It's kind of messy. That's definitely something we want to do, and we will try to get that there. We can definitely get the list. That's something that's very, very easy. We can get the training and validation lists that we used on the repo. And then, like I said, the longer term goal is to get the actual training script available. So all the pre-processing, all the training script, that is just going to, unfortunately, it's just going to take some time to do, but we're hopeful we can have that in sometime this summer, hopefully.
Yeah, but I think, yeah, part of the question is on the list, I think. So that's probably what I think the community wants to also reproduce or use the same data for other purpose. So probably that's the question. Yeah. And so as you said that there is this issue, I mean, there is this on GitHub, we can make issues and ask for things to fix, because there is one of mine, but yeah, I will use this opportunity later, because I tried to model protein RNA and there was some problem that I couldn't fix on my own, but I will I will.
Okay, yeah, sorry, I'm sorry. I need to look at
but yeah, but that's to everyone, like that's the place. I think there are some issues. Other people also try to help other people. So if not Frank, then someone help. So that's, I think always it's so great to share these issues with other people, because sometimes I reply also on other reposts of other people when authors don't reply, and I know how to say it. And to finish maybe this quick session, there is yet another question from Jam Johnson. I can read it. Did you calculate TM score of your models?
We did. So we do normally, not for this data set. We did not, unfortunately. I just put this together recently. So I didn't calculate the TM score for this. I don't know. The RMS, we did look at RMS for this, and it was just way too noisy. That all the models basically had high RMS, I think. But I mean, I think even the best server had relatively high RMSs. I didn't calculate TM score for this, but we did look at TM score for the for a validation set later, and that seems to track fairly well with the LTD, with the other metrics, basically. All right.
Okay, then Risa. Sorry.
Hello.
Hi.
Thank you for your talk. I have a couple of quick questions. So I tried this early on when it was just released, and sometimes if the protein was a dimer that is binding the DNA,
Yes,
the two monomers would completely overlap on top of each other. And
yes,
there was some discussion on GitHub about this. I was just wondering if this is already fixed or not.
it should be fixed in the current version, and I'll talk about that when I get to complexes in just a second. Yeah, that was a problem with the early versions of the network, is that can I get to, because I'm going to talk now about the next about the protein, the complexes. That should be fixed
All right.
now.
Yeah. So my other question is, can we provide an initial structure for the protein and the nucleic acid to kind of kickstart the prediction?
Yes, so that can be done through the templating track. So there's not right now, the shell script itself doesn't have support for that. I don't think. I'd have to look at that and see. But basically, the model itself, if you can contact me offline, I'm happy to set up some of the methods for that. The way we've done this previously is to feed that as a template itself, to give the structure as if it were a template, right? So both the nucleic acid and the protein, as if it were a template. It's sort of, yeah. And that can help drive the structure in some cases. It's sort of interesting. The network, the way it uses that information can be somewhat tricky in that sometimes, especially, sometimes it will ignore that information a little bit, but that's how we basically give the network a starting point. Yeah.
Thank you.
Yeah. All right.
So maybe the last one, if either of the protein nor RNA have any model structure available in the database, can we still detect the complexes with a good confidence score? So
yes, sometimes the network will be able to predict things. I mean, I guess that's what some of these, whoops. Again, well, this is for RNA, but for complexes, we'll sometimes get that. I mean, again, if the MSAs are deep enough, and if the folds are sort of, again, it's somewhat unfold, the folds themselves are simple, the network will can oftentimes predict structures accurately, even if there is no homologous structures that are available. And I'll show examples of that in a second. Yeah, I think because there's a lot of questions about complexes. Let me get on to some of the complex work. So the next thing that we looked at, right? So the other thing our network predicted was protein nucleic acid complexes. And again, at first, we were happy with these results. It looked reasonably well, but then we took a closer look at the data, and the data was somewhat problematic. And specifically, this idea of DNA sequence recognition, the model seemed to have a really tough time learning. This is an example of this structure 7127, and you could see the posited structure, predicted structure, binding mode is dead on, right? It really gets the binding mode quite right. But if you look at this, the original protein itself has very high specificity for this particular nucleic acid sequence, but the model predicts this, basically the DNA, it just rotated 180 degrees. And so, we also then looked, so this was, again, one of the things we really wanted this network to be able to do was to predict DNA binding specificity of proteins. And when we looked at this, this is on a data set. I'll talk more about this data set in a bit, but basically a data set of transcription factors and their DNA binding sequences. We find that the network essentially wasn't able to distinguish between those. So what I'm showing here is basically what we're using to stand in for whether or not things bind is just the model accuracy between chains. And what I'm showing is basically the binding DNA. So these are the same exact proteins compared to DNA sequences that it binds, DNA sequences that it doesn't bind. And you can see no separation between these. So what we ended up doing with this point, oh, shoot, sorry. Yeah, I was like, look, we have to be able to improve this. And so we made a couple of changes to the network. And so it seemed like, I guess, essentially the network was learning kind of two things. It was learning to kind of memorize certain sequences that were very prevalent in the binder data. And then it was also like learning that basically most of the time it should just stick the protein in the middle of the DNA, because most of these crystal constructs that it's learning from are like 8 to 10 bases with a protein right in the middle. And so the network was learning that. And so what we needed to do was to trick the network. And so we did two different things. First of all, we just did random padding, and we padded between 0 and 6 base pairs on each side. So that potentially can introduce new binding sites, but every time it sees an example, it's giving it a new binding sequence. And so I'm hopeful that the network rarely includes this padding. And this padding was only done to complexes that made at least 3 base-specific interactions as well. So the odds of it introducing a new binding site, I think, are relatively low. We also explicitly trained the network to discriminate negative and positive sequences. And this is something that we're doing that we also do for protein-protein complexes, and I kind of glossed over it a little bit, but basically the idea is that we train the network so that when it sees positive examples, it has to predict the complex, right? When it sees these negative examples, so DNA binding proteins and DNA sequences that they don't bind to, we do this by just randomly mutating all of the base-specific contacts. Again, and the hope is that for the vast majority of cases, there's not a DNA binding site. You're still not, a protein binding site still in the sequence. And you basically train them, at least in this case, to move to the right on this prediction. So you don't care what their overall structure is, and you just say, I want you to have high uncertainty between your models. And then the last thing that we actually did to this network was to specifically, so this was using kind of uncertainty across the interface, but instead what we're adding to this is this new head to the model, this probability of binding head that actually separates this task. So uncertainty is still just coordinate uncertainty, and now you actually have this binder head. And we actually use this for protein-protein interactions as well, and that seems to help somewhat at predicting protein-protein interactions as well. And so now, when you look at this, you can see we're now able to really have some knowledge of DNA binding specificity. And you can see, so this is the initial curve, and then on the bottom, you can see there's definitely separation. So there's a lot of binding sequences that we don't recognize as binders, but now things that we give high confidence to, the vast majority of them are actually DNA sequences that bind to the protein. And you can see we're plotting this in terms of transcription factor families. And you can see, again, this varies quite a bit. Homeodomains we do really, really well on. Some other families we do less well on, and there's reasons for this that I can get into if people are interested, I think. Yeah, go ahead, Rachel.
Quick question. Your probability, have you guys checked out if it correlates to binding affinity yet?
We have not. We've been so in other contexts, we're really interested in binding affinity. The problem with the data that we're using for this, right? We're using, so basically, I'll get to this in a second, but we use CISBP. Data comes from different labs, and it's not totally clear. It's all enrichment factors. It's not totally clear how well normalized this data is. So we haven't. Ideally, what we would like to do is to have something more quantitative coming out of the network. So maybe like quartiles of these binding affinities or something like that. Right now, we're doing this. So basically, maybe do I show it in a bit? But basically, this is how we are processing this CISBP data, is that very high things are positive, very low things are negative, and there's a mid-range where we just say that we don't know, these may or may not be binders. These are weak binders. We haven't looked at this. The thing is, in parallel, we're doing this with some protein-protein binding data, and there we're actually trying to make use of quantifying basically the these binding affinities, that they're all, so we have a subset of it, I guess, that's more uniform. And so there we're going to try to look at that. Right now, we haven't really looked at this, and I don't think, my guess is there's been very, very little correlation, if any, there. Yeah, you?
Okay, yeah. So yeah, this is a very nice work. But I just wondering, so a few years ago, Brandon Free's group proposed the DeepBind. So I guess probably you also know the work, right?
Yeah,
so
yeah.
for their work, they essentially the input is still the sequence, but they didn't consider protein structure.
Yes.
Again, I mean, based on their experiment, they said that their model, although for each protein they have one model, they can already identify the DNA binding preference or RNA binding preference of the protein. So I'm just wondering, did you try to compare with that method? So I know it's a little difficult to compare because for each protein they have one model, but for you guys, you have one model for all the proteins. So
exactly, and that's the goal of this. So I would say not specifically that model, but we've looked at some. There's a wide range of literature of sequence-only models for one family. And yeah, our model is definitely not as accurate as those models, sequence-only models. But our goal here, and you'll see this in a second, our goal here is to train a model that can look at understudied transcription factors or predict novel transcription factors where you don't have that sort of data. That's really, I think, I mean, the problem is these family-specific models do really, really well, are very, very highly accurate. And so it's really, it's difficult. I think the best we're going to do is to approach that level of accuracy. But the idea here is something that's cross-family, right? Something that, I mean, that does two things, that can go, again, understudied families can predict DNA binding specificity patterns, and then also something that gives you the three-dimensional structure of that binding mode. So I will fly through some of this data here at the end. So the structure prediction, again, looks very, very good. So we make these changes to the network. Structure prediction looks very, very good. In terms of, again, this is all, same plot as before, sequence identity of the protein. You can see that there's a bunch of cases where it's completely novel in terms of binding mode. And then these examples from this cloud on the right, basically these points up here, are shown on the right. Both protein DNA and protein RNA structures are shown. We can do this for, again, a lot of these DNA binders or dimeric complexes. Getting back to the earlier question, one of the early problems in the network was that things were always modeled as monomers, right? And so following what we do with proteins, if you have a homodimer binding to DNA, it would, you basically would train it so that it could find either binding mode, and it was rewarded for that. But it turned out that led to this problem where it wanted to hit both sites. And so the DNA and both chains would lie on top of each other, basically. And so the newest version of a model has been trained with, if the structure is a homodimer, it's trained to predict the homodimer structure. And so now this does a really good job at predicting these homodimeric complexes as shown here. And then the last thing that I wanted to show, again, so I think one of the things is like, there are methods for predicting RNA, right? There are methods for, I mean, you can build B-form DNA, and you can predict with AlphaFold very, very accurate structures. Is the network doing more than that? And I think yes. So one of the things, I'll skip this plot on the left. We basically tried that experiment, and we find that that docking really has a difficult time with predicted models finding the right binding mode. But I think more importantly on the right, I'm comparing some predictions of the network with both protein, nucleic acid, and complex, and individual components separately. And what you can see here, this DNA, right? This is curving. It's this homodimer basically induces kind of this curvature, this bend to DNA. In this case, if you predict DNA alone here, you just get something that looks very much like B-form. And you can see it's very different. Whereas if you predict it in complex, it gets that overall bend of the structure. Same thing with proteins. Again, so proteins are very, very well predicted by both AlphaFold and RoseTTAFold. But like in this case, for example, there's a 3-domain protein here. If you predict it, it gets the 3 domains right, but it just doesn't get the orientation of this 3rd domain correct. But if you model it with the DNA, it now realizes, oh no, this helix, this terminal helix heel is actually interacting again with the major groove of the DNA. And so it gets the structure of this correct. Again, you can see an overlay, the structure predicted alone versus the structure predicted with DNA. It's able to just twist that last domain. And so we think like these types of things would be difficult to solve by docking, right? the structures themselves aren't right. They can only get predicted right if you actually have, again, the DNA present in this case. Yeah, so where we're going from this, and I think this ties back to an earlier question that people said, how do we make these models better? I mean, the problem is, look, we see accuracies that are, generously 40%, right? Where 40% of structures, and I think it's still a useful tool because the network really does know when you're in that 40%, but it's only 40% of the time compared to, again, protein monomers where it's 90% plus of the time that you get really very accurate predictions. And so the reason for that, I think, is we're just data limited. Like I showed, we have 1/20th of the effective data. And so what we're trying now, and I think really our idea of getting more data is to be able to make use of indirect data in terms of fitting this network. And so what we've now been trying to do is to take, I've showed this sort of transcription factor, but there's kind of large databases of transcription factors, what DNA sequences. So basically transcription factors and 4 to the 8th kind of sequences against 4 to the 8th sequences, 8 nucleotide patches. That's like, I mean, in some sense, it's a lot of information. It's a lot of different sequences. On the other hand, it gives you a single value and enrichment value for each of those sequences. But the question is, can that data help the network in some way? And specifically, can that make up for this lack of training data that we have? And can this help structure prediction? And so we set up training with this network. I will fly through this. Yeah, so basically the way that it works, we have our structure prediction, but we also have this binder head. And so what we do is we take our normal training. We normally see a complex, a crystal structure. We predict the structure of it. We see sort of a data point from this binding data, and instead we don't care what the structure predicted is, but we say this binding head should either bind or not bind, depending on whether it's a positive or negative example. So we only constrain the binder head. And then when we train this, it's about 1/3 of the examples are these transcription factor, is this transcription factor data. 2/3 of the example are the original structures, crystal structures that we're training on. And so what do I want to say? I'm going to jump through some of this. So, okay, basically, maybe I won't jump through this because there's only 3 slides left. Basically, what we see is we see some ability to predict binding affinities. And so I'm showing, so basically what we do is we split these families into training and validation, right? And so of course, on training data, we see an improvement. Homeodomains are something that was already predicted well. And now we get like really great separation, but this is in our training set. Zinc fingers and these AP2 structures, these are ones that basically had 0 separation initially, that both of these plots were on top of each other. And now we're seeing the zinc finger is in the training set. We're seeing some separation. Sorry, let me move my zoom window out of the way. We're seeing some separation between these two, but what's nice is this AP2, this was in our validation set. The network didn't see any of these examples. It's a sort of non-standard binding mode where you have, again, a beta sheet rather than a helix, making the protein DNA interaction. And we're starting to see some separation here. That said, there were problems here also with dimers, and we noticed some really weird behavior where some structures, not a lot, but some structures were predicted where basically the DNA, you had 2 binding sites on the DNA that just overlapped one another. And this had to do with the fact that for 2 things with this data set. So first of all, this data set has a lot of things that aren't homodimers, but it's clear from the data that there are 2 copies of the protein that are interacting, and the highly enriched sequences have 2 copies of the protein that are interacting with them. And then the second thing is that because of that, you get these symmetries in the positive set. And it seemed like the network is learning a lot of those symmetries, unfortunately, that it's learning certain, if you have a certain symmetry, right? So you have, again, a binding site this way and a binding site this way, or 2 binding sites in a row running this way, that's very indicative of positive binders. And so what we've done now is set up training so that we maintain the symmetry. So basically, for each positive example, we generate negative examples that have the exact same internal symmetry of sequence with them. And then also cases where there's high confidence of 2 binding sites, we model them as homodimers. Even though they're not maybe binders, they're not really interacting with each other, we model 2 copies of the protein in the simulation. And we do this by basically running with both all of these examples with both monomers and dimers. And then we look at the predicted error basically between both chains, and we see if that's above some threshold. So this is like ongoing right now. I'm showing some of the data here. You can see here in terms of precision and recall of our validation set, you can see that our precision is increasing fairly significantly. We go from 50% to about 70% precision. The recall stays flat, maybe drops a little bit. This is promising, but that the most promising thing is that if we look at the all-atom LDDT of the protein nucleic, of the protein DNA, this is protein nucleic acid generally, unfortunately. So this is, you need to specifically look at the protein DNA, but we see some improvement here. It's pretty subtle, for about 0.66 to about 0.67. So very subtle improvements, but we're happy and we're hopeful that this, again, it's learning something more about, again, fundamentally the energetics of proteins interacting with DNA. And so there's a lot left to do. So this is like ongoing, this training run, basically. I haven't done the full analysis of this yet, but I'm cautiously optimistic that from this plot on the right, that this is useful. There's a lot more data. So where we're going with this, there's a lot more data available from other papers and databases, and we've started assembling that data together. Yeah, also there's, again, a lot of DNA binding protein engineering studies that also have collected experimental data, both DNA mutations, but also protein, also transcription factor mutations. We're just in the process of collecting that data and using that data to train the network in the same way. And then finally, we're kind of curious about distillation data sets. So for this data, as you can see, most of these structures, Rosetta, well, this is the bad example, but many of these structures, Rosetta predicts reasonably accurately. And so one idea is to actually implement this distillation data set. So take these highly confident transcription factor structures and use them as additional training points as well. And so we're in the process of kind of setting up all of these as well for the next rounds of experiment. We want to potentially, again, memorization. There's a lot of data here, but the number of positive binders still isn't like massive. I think it's like 10,000 or something like that, which is good, but not spectacular. And so one idea is we may need to do the same sort of DNA padding tricks. And then if this works, I think this is a formula for potentially making use of kind of high throughput screening data in a network like this. And so I know there exists lots of data for protein RNA complexes. And so I think this, we haven't looked at this yet, right? We haven't done anything along this way, but I think this would at least give like a roadmap for the way to make use of that data in training. And I think, yeah, with that, that's the end. And so this work was done mostly by myself, Brian McHugh, and then of course Ming-Young Baek, who was again really the main person who spearheaded RoseTTAFold. And so with that, I'm happy to take questions if there are any more.
For this, it was very informative, and I think we all learned, and we have more questions in our chat. So where was the last one? Yes. Is the model able to benefit from potential symmetry with dimer binders?
So we have not actually specifically we've not specifically looked at that. You're saying running with a symmetry mode. And one of the reasons is you don't get, when you're doing dimers, you don't get any savings in running it in sort of symmetry mode versus not symmetry mode. And so we've just been running these basically sort of like asymmetric dimers. One of the other issues is like in the transcription factor data set, sometimes the DNA sequences aren't perfectly symmetric. They're very, very close, but like the end positions vary a little bit. And so it breaks symmetry a little bit, even though the binding itself is symmetric.
So maybe another one from the chat. Are there any successful cases in your test that have a new binding mode different from the training data set?
We honestly, we have not, I would say we have not looked deeply into that yet. We're still trying to just dramatically improve the performance of this network. I mean, it's a good question. That's something we definitely need to do.
Okay, Yuli, you can.
Yeah, so I mean, this is a very nice work and a great talk. So I'm just wondering, so you mentioned that you want to collect more data. I'm just wondering whether it's possible to try to combine the data from MD simulation to the current data in the PDB database. Do you have any idea of doing this kind of combination, or do you think it's useful to try to combine MD data? Yeah,
I mean, that's a good question. That's something that I know I definitely have come across that people have been doing this. I think especially for protein small molecule interactions, where I think there's even more of a data shortage problem in terms of that. I am not, I mean, it's one of those things I think you would just really have to try and see. It's difficult. Like, again, so the idea would be maybe it's giving the network some information on like energetics, right? It's telling the network that you need to predict structures that have, you know, reasonable energies. That's, yeah, I mean, that's, I don't know. I think it's not, I mean, it's
I
a good
agree.
idea.
Probably we'll have to try.
You just need to try it, I think,
Yeah,
and
probably.
see. I will say a little bit, we've started looking at this for sort of protein small molecule interactions, but it's still super early and it's really too early to say.
Yeah, and it's always very computational, I mean, demanding to get a lot of MD simulation data. Yeah, that's true. Yeah, great. Thank you.
Yeah.
Okay, so maybe the last one. So the question is pretty much about additional tokens that could be added eventually to model modifications, both for RNA and proteins.
Yeah, I mean, it comes down to like training data. So in terms of adding new tokens, right, you would need to see enough instances of those tokens. And I think at a certain point, for certain modifications that are just not seen super often, you would probably you would probably need to have something more general that could really handle things at the atom level, right? I just think it learns a lot from canonical amino acids and canonical nucleic acids because it sees thousands and thousands of examples, right? I think you need, you know, if you're looking at, I mean, I'm just trying to think, I guess maybe common glycans you could do something with, there might be enough data there, but I think for most modifications, there's just not enough data to like add them as a new token and have the network really be able to robustly learn their types of interactions.
So maybe one more for Marco, because now I see it and it goes like kind of broadly into like super, you know, future. Is there you have an ideas to have, you know, these tools that will work on single sequence without using alignments?
So I think that's a really important question. I mean, for me, at least personally, it's bothered me that native proteins need MSAs to fold, that like you don't, you know, nature knows how to fold these proteins correctly with just seeing a single sequence. I would say though, I think from the training experiments, I think that what we found is that very small versions of RoseTTAFold really learn like the MSA information. So I think there's like two aspects of the network. The MSA information kind of broadly gives you the fold, and then the rest of the network, it's learning something about energetics and it's snapping everything into place. And what it seems like is very, very shallow networks can learn that sort of the MSA information. And so a network that's very small, very shallow can get 90% of the performance of RoseTTAFold. And you need all of these extra layers, all of these additional parameters to really get like the physics of that right. And so I just wonder if like you just need to actually go completely and determine that three-dimensional structure from a single sequence, if you just need like a much larger network. And then of course training becomes challenging. I don't know. It's something I'm very interested in, but I have, I guess, guesses, but I don't really know what the best way to do that is. I think probably, I think potentially a more expressive network, potentially giving the network more information on energetics. Like we know stuff about energetics. We know proteins don't clash, right? We know proteins like to make hydrogen bonds. If that information could be provided to the network in some way, maybe that would help sort of drive things. But no, I don't know. My guess is maybe a bigger network would and specifically trained with very shallow MSAs could potentially get there. But yeah, I don't know. I mean, I will say the one thing I will say is people have tried training like this RoseTTAFold's architecture with single sequences, and it really doesn't learn. Like it doesn't help the ability to predict single sequences.